I think that's quite obvious by this point of time in the course, because most biomolecules are nature's robots. When I'm talking about biomolecules, mostly proteins here. And because it's only their specific three-dimensional structures, uh, because of their specific structures is why they are actually able to perform functions, right? So if you have a machine which is doing some function, only specific shapes of certain machines will be able to do that function. So let us say you have a machine to crack a nut, right? You had different kinds of fulcrum load and I forgot the third thing, fulcrum load and does anybody remember the three things that are there in any kind of a simple machine? Anybody listening?
Yes, sir, we are listening.
So what are the three things in simple machines? There's fulcrum, load, and effort, right? So basically,
Yes.
depending upon the placement of these three in space, you get different kinds of machines. So if you place the fulcrum at the end of the load and the effort, then you get something like a nutcracker, right? Where the effort is in the middle. So basically, shape of machines is very important to their function. So when we talk about cells, there are many shapes which are existing, and all of these shapes can be talked about in an umbrella term of structural data. So when we have so far what we have talked about genomes, we have talked about DNA and RNA sequences, we have talked about ontologies, we've talked about literature as well. Um, I haven't talked about gene expression. It's an advanced part of the course. We have talked about pathways. We have talked about how the different organisms could be organized. But then all of these things, for example, DNA and RNA themselves have some structure. Proteins are organized in motifs and families, which are inherently determined by their structure. Two proteins or many proteins can come together and interact. Proteins themselves have structure, proteins have the sequence, right? And a lot of these proteins are then interacting with some chemical entities or small molecules. So when we specifically talk about um, structural data, it's a composite of sequence of the biomolecule. You talk about the structure of this biomolecule, and we talk about the inherently the structure is coding for the function. Now, in order to talk about these things, whenever you're talking about sequence and structure, you're essentially talking about energetics. So basic physics and chemistry principles of how the different monomers are coming together to form a polymer. And then when we're talking about function of a structure, we're talking about how that particular structure is going to move in time or how it's going to be dynamic. So you're talking about whenever we are talking about structural data, we're talking about its energetics and its dynamics. So how does it look for proteins? In the case of proteins, you have the sequence of proteins, which is basically nothing but amino acids, where you have your peptide backbone. So in the unfolded form, it's just a chain of amino acids. It's mobile, but it's inactive. When it gets folded, it forms a specific structure, which is ordered. Basically, it has each of the amino acids have a relative order with respect to each other in 3 dimensions. It is stable. Most of the amino acids don't change with respect to each other, but it is dynamic. Some of them might change with respect to each other. So for example, if there is a loop here, it's possible that all the amino acids here are always going to be together. All the amino acids here are always going to be together, but these amino acids, three amino acids here, might actually change their relative position. And then once you have this structure, which is stable and dynamic, this can have specific conformations inside the cell. So for example, here you're seeing two phase separations. So maybe one part of the cell is aqueous, the other part of the cell is non-aqueous. It could be polar, could be non-polar, say the membrane. And depending upon where this protein is, its dynamics are going to change. and appropriately its function is going to basically be visible. So now in daily life, when we use machines, um, these machines have some functional structure and moving parts. So let us say we use a machine like a bicycle, right? Everybody has seen a bicycle. Now, so far, what we have talked about with genome sequencing, genome annotation, it essentially what it's giving us, it's giving us a parts list, right? So we have a sequence of an organism, and from the last lab and from last module, you can annotate the sequence with different kinds of features, such as genes. And each gene actually is a part, right? It's a part in the entire organism. So basically, this is what you get when you are looking at the genome. But we don't actually know how these parts are put together, right? So this parts list is not enough to understand how the bicycle works. So what we actually want from structural biology to give us a full spatio-temporal picture of how the parts are individually placed, right? And how they are coming together in the form of the entire organism. All right. And once you are able to do this, once you understand how this organism has come about, then you can also manipulate. You can also engineer different things, such as you can engineer some drugs, which is called drug design. So maybe let's say if you have a bicycle, in the bicycle, if you understand how the bicycle is put together, maybe you can put a light here, right? That could be a design. So you similarly you can do it for proteins. If there's a protein which is put together in 3 dimensions, you can specifically decorate that protein with a drug molecule, which is a small molecule. If you know how the bicycle is put together, let's say how a protein is put together, then you can even understand what happens to this particular protein. Let's say when the bicycle, I don't know, when the chain comes off, right? So because you know how that where the chain goes, you can fix the bicycle, right? So the same way you can diagnose what's happened to your organism if you understand how all the proteins are coming together, how the molecules are coming together, and you can diagnose things, what's wrong with the organism, and also you can fix it by appropriately doing appropriate intervention. And at the end of the day, if you understand how all the parts of the bicycle are put together, you can actually manufacture more bicycles, right? So this entire thing is directly analogous to what happens inside cells. You want to design drugs. You want to understand how proteins are not folding properly or are not interacting properly and causing disease, and then reset them. And once you understand how proteins are put together, how macromolecules or complexes of proteins are put together, you can even start to manufacture these things using biotechnology means and or bioprocess. All right. So when we are talking about structural biology, the most the the most important part is, so you can get the structure, you can get some 3 dimension, but at the end, the most important thing is the function. And this function is based on the conformation that a protein takes. So what do I mean by that? You have a protein, which is basically a string of amino acids, which is sort of unfolded. It is disordered, right? And then it travels this energy landscape. So it has all possible combinations of folding, right, in 3 dimension. And then whenever it gets folded in a position in a shape where the total free energy is minimized because of many kinds of molecular forces that I'm going to talk about later, you get a more compact or more ordered structure. So the key concept in structural bioinformatics is this idea of this energy landscape, which essentially allows a protein to fold and get a specific stable but dynamic structure. And now when this folding process is happening, you also have, as I said, you have some time that is basically involved here. So going from a totally unfolded state to some folded, semi-folded state, it's called a molten globule state, which is compact, but it is still disordered. Then it sort of, this molten globule state, it's has more combinations, it folds a bit more, it searches more combinations. This takes about 1 millisecond. And once it has sort of crossed this barrier, then it becomes its native and compact stage. So like this, protein can have multiple conformations, and these multiple conformations can be determined by whether the protein is acting by itself or whether the protein is acting along with in concert with some other molecules. This could be small molecules as shown here, or it could be actually another protein also, or it could also be dependent on exactly where the protein is present inside the cell, whether it's inside an aqueous compartment or a non-aqueous compartment, whether it's if you talk about non-aqueous compartments, whether it's in the mitochondrial membrane, let us say, or it is in the ER membrane or the Golgi membrane, inside the nucleus, outside the nucleus, in the nuclear membrane, cellular membrane. So, right. So all of these things essentially affect how this protein is going to get different conformations or shapes. So the outline for today, I'm going to first give you an overview of structural bioinformatics in terms of its motivation, its goals and challenges. Then I'm going to talk about fundamentals of protein structure, very basic. And then I'm going to end up with how you are actually able to visualize protein structures and what information you can get out of this visual inspection. So the first motivation is that you can look at how the protein is coming together in 3 dimension, right? Specifically. So with a detailed understanding of how the different molecules, I mean, in the amino acids, it provides you a structural context in terms of why is it that certain amino acids in a multiple sequence alignment are conserved, right? When you are seeing how these conserved amino acids are folding, are coming together in 3 dimensions, then it will give you some kind of a functional insight. The other thing is that computational modeling of these functional interactions. So whenever you have conserved amino acids interacting with each other, then this would give rise to some kind of a functional interaction, because this is important for the function of the protein. That's why it's conserved, right? And these functional interactions can help you understand what exactly the protein might be doing and how it could be regulated, or what will happen if there would be certain mutations in these conserved locations. The second motivation is that the collection, just like the number of genomes, the number of protein structures is increasing. So there were about 160,000 protein structures about 2 years ago. And since the advent of AlphaFold, these structures have increased many fold. Right. So this is the stats coming from there. So given that you have these structures, now what you can do is you can in silico, right, uh, start to make more try to make comments about other structures, right? So structural genomics is another subfield of structural bioinformatics, allows you to specifically come up with structures for other species. So for example, if you have structure for one species, uh, and you have a new species come up for which you do not know the structure, but you can do multiple sequence alignment and you can see which areas are conserved. And you can then predict that most likely the conserved areas will be folding in a similar fashion. You can use this information to fold this completely new protein in 3 dimensions. And the other thing is that theoretical and computational predictions have been valuable. And because of the computation power that we have currently, we can actually theoretically solve much bigger and much larger protein structures. So therefore, it's one of the motivations behind actually pursuing structural mathematics. So the key motivations is sequence of a protein gives rise to its structure, which gives rise to its function. So if structure determines function, so understanding the structure will help us understand the function of a protein. Structure often is more conserved than the sequence. So if you are looking at structures of proteins which are performing similar functions, most likely they would be much more similar to each other in shape than in sequence. And the third motivation is that the structure is encoded in the sequence. So now we know that it was a 100-year-old unsolved problem, but now it has been solved with deep learning, that with structure, with just this information of the sequence of a protein, you can actually predict and get an accurate information about how it will fold in 3 dimensions.
Sir, I
So
have
now
a
what?
question. Sir,
Yes.
Sir, you described that when this protein will be inside the nucleus or outside the nucleus, when it will be in mitochondria, then its a structure like changes. So
Yes.
how can we say that its a structure is totally determined by this amino acid sequences and nothing else?
Yes. So you get what's called the most basal native state of the protein. But what I was talking about when the protein is in different locations, its conformation changes. So its relative parts of the protein change. So this is the dynamics of the protein. So if you, for example, imagine that your protein looks like a knife, okay, or looks like a scissors. So what you can imagine is that maybe in the cytoplasm, the scissors is going to chop much faster, right? But in the nucleus, because the environment is not favorable, so the chopping motion of the scissor is going to be slower. Do you make sense? Does it make sense? So the
Yes, sir.
flexibility of the protein is going to be slower. So the conformations that would be allowed in different parts of the cell would be different, but the major shape of the protein, that is the protein being a scissors, right? That cuts is going to be relatively fixed, relatively stable. So these are, so when you talk about structures, we talk about stable structures and not how they would dynamically interact in the environment. But you do remember that whenever we talk about a structure, it's an average representation. It's not the it can still relatively it can move around. Does it make sense?
Yes, sir. Also, if you can describe the second point, like structure is conserved, does it mean the same?
Structure is conserved essentially means uh, that proteins which are performing similar functions, right? Even evolutionarily distant uh, proteins. So for example, if you take a protein from E. coli, which is a bacteria, and you take a protein from humans, right? So one of these proteins would be ATP synthetase, right? So ATP synthetase would be
Yes,
found
sir.
in many bacteria and would be found in humans as well. So this is extremely large evolutionary distance, right? You would see
Yes,
that the
sir.
structures are very similar, although there might be some sequence differences, but the structures are going to be very similar because they perform at the end of the day the same function.
Yes, sir, but their structure is different, I guess. Like in mitochondria, all the mitochondria have same, but in bacteria is like, I guess it is different. In chloroplast, it is different, I
Yes,
guess, sir.
but that's what I'm saying. So the major, so that's what so think about it in this way, because it's essentially channeling ions, right? So in order to channel ions, in this case, hydrogen ions, right? That's what ATP
Yes, sir.
synthetase does, right? So it has to channelize ions. So there was only one kind of structures which will allow you to do that. So in spite of the fact that there would be mutations, these large, let us say modules of the machine are still going to look very similar. So if I were to give you an example, a bicycle made by Atlas, it's an Indian brand, right? I don't know if people still buy a bicycle from this company, is going to work like a bicycle, right? And similarly, you have like a lot of these foreign brands coming up. Even Mercedes makes bicycle. I think one you in UP, Akhilesh Yadav, he probably had a Mercedes bicycle, right? So it's still a bicycle, right? So the machine is the same, but it looks different. So when I say
Yes, sir.
structural conservation is there, your goal as a bioinformatician, structural bioinformatician, would be to talk about the function, right? And in order to identify proteins with similar function, this statement tries to say that you want to look at the shape of the protein and the major grooves and major patterns in the shape rather than just the sequence. The sequence, individual parts of the two bicycles must be very much can be very different, but when they are put together, both of them do the similar function. Does it make sense?
Yes, sir. Sir, so are we able to like find using like computers that what is its function also?
Uh, Not exactly, but you can say how it probably moves, and you can then try to study how a structure would be interacting with other things, because the function of the proteins is not going to be in isolation, right? So a protein can either be a structural protein, right? That it forms this like a building blocks of the cell. So if you know the structure, then you can put together the structural protein like actin in polymers, right? And like multiple actins can be put together in a specific way, and you can try to find, okay, probably this is how its function would be. It will be put together like this. If it is an enzyme, so then it will have a substrate. So you will see, okay, once you have the shape and you add the substrate to the protein, exactly where in the protein it's binding, right? And once you know where in the protein it's binding, you will see which amino acids are interacting with your substrate, right? And most likely if it's an enzyme, so enzyme would be removing a chemical entity, adding a chemical entity, or changing the shape of the substrate, right, itself by breaking it. So those kinds of things you can understand once you have the structure.
Yes, sir. Yes.
Okay, so I can actually talk about those things. So the first thing to do any of these things that I described is to actually visualize the protein. And you can visualize the protein in space and time. And this is one example of how a protein looks like. The next thing that you can do is once you are having a structure of the protein, you can analyze the differences and similarities between different types of proteins. So for example, here there is an ATP synthetase, here is an ADP synthetase. So these are different proteins, and you see that proteins which are performing similar function. So this is called a PCA plot. It tries to plot different features of a protein of any data set. In this case, it's plotting different features of a protein in 3 dimensions. You see that features of three-dimensional features of proteins which perform similar functions, they are falling together in space. The other thing that you can do is, so here you were doing analysis of proteins for a lot of three-dimensional functions. The other three-dimensional features here, you can do specific comparisons of two proteins. So if you see, this is a protein which is um, myosin motor. So it's a motor protein. So this protein actually walks on myosin fibers. And here you can see that there are two structures. So one structure is in yellow, another structure is in yellow, and one structure is in yellow, and other structure is in red, and it has been superimposed over each other. So these two protein structures have been now compared at the very amino acid level of the structure itself, right? So you can actually compare two proteins, and this is very important when you are trying to compare two proteins from different organisms, or if you're trying to compare a wild type protein and a mutated protein, right? You will put them together and you will see exactly how the structure changes and predict exactly how the function will change. The other thing that you can also do is, given the structure, you can predict the interactions of this particular structure with other molecules. So it could be other proteins or it could be other small molecules. In most cases, it's small molecules because this is a computationally tractable problem. So you can solve it on a computer easily. So once you can actually predict interactions with other molecules, you can also design interactions. So for example, you can design new drugs which bind to the protein much better, or which inhibit the protein's movement in a much more better fashion than existing molecules. So for example, if you are creating a drug, this thing is mostly used in the case of drug design, where you want to inhibit a function of a protein, which, let us say, a protein which is there in tuberculosis bacteria. It allows the tuberculosis bacteria to hibernate. As we saw, it hibernates with certain motifs, right? So if you
Yes.
block the protein, the transcription factor binding to the DNA by a small drug, you design a drug, then you can essentially stop these hibernation of proteins because the transcription factor is never going to bind, and therefore the genes are never going to be expressed. So the hibernation will not happen, right? So you can do things like this. You had a question, Ratnesh?
No, sir.
So these were the basic goals of structural bioinformatics, but there are certain challenges in achieving these goals. So one way would be to do protein classification, because in order to actually talk about the function of a protein, you need to have some prior information about some protein classes, right? The other thing is that it's the problem of structure prediction from sequence. Although it has been solved for some basic structures, it is still an open problem. How do different molecules come and bind to the protein is also not a straightforward problem. So this is the challenging problem. Once a small molecule goes and binds to a protein, whether it would be a stable binding, it would be a non-stable binding, unstable binding. So this will be important for drug design. The other thing is once you have the structure of the protein, how does the structure actually move around? How does this, what is the dynamics of this structure depending upon where in your body or in the cell this protein is available, because depending upon the surrounding of the protein, the physical forces acting on it will change, and that will change how the motion of the protein is there. You can do prediction of simple physical properties such as stability, binding affinities, and you can also design the structure and function of new proteins using synthetic biology. And these would have applications in biology, medicine, agriculture, and industry. All right. So now I'm going to talk about some fundamental ideas of protein structure. I'm only going to talk about the three main ideas. So the first main idea is proteins, just to remind you, are made up of amino acids. Amino acids are monomers. They have this kind of the nitrogen amino group here and the carboxyl group here. And they have a side chain which can be having a large size, or it can have different charges, or it can have your rings or lack of a ring. And these monomers come together and they form a primary structure, which is just a chain, which folds into a secondary structure that actually folds into a tertiary and quaternary structure, what you actually see. So when we talk about structural properties of proteins, we are mostly talking about everything which is not a primary structure. So secondary structure onwards, this is what you talk about in structural bioinformatics. So when you talk about structure, secondary structure, uh, one of the main ideas there is to predict the two main classes of secondary structures, which is alpha helixes or beta sheets, and they depend on how the bonds between two amino acids are rotating. So the bond between the first bond, the C, and the first bond between your the peptide. First part of the peptide bond is called the phi angle, and the second part of the peptide bond is called the psi angle. So depending upon the orientation of the phi and psi angle, you can make a plot like this, plotting the phi and psi angles in your protein of interest, and depending upon the relative values of these phi and psi angles, proteins can take certain secondary shapes. So we call these secondary structure shapes because these secondary structure shapes can be described on a paper sheet, and therefore these can be of two kinds. It could be an alpha helix or it could be a beta sheet, and this can be basically seen through these kinds of Ramchandran plots. So when we talk about three dimensions, like looking at protein in tertiary or quaternary structure, basically in three dimensions, how do proteins actually look like? So this is actually how a protein looks like. Can you tell me what are these molecules? The protein is somewhere here. There is the protein and there is something else also here.
So is it water?
Yeah, it is water because you have one large oxygen with two hydrogens, right? So actually the proteins are stable and they are hidden in water. So if I remove the radius of these molecules, this is where your protein is hiding. So most proteins are actually closely interacting with water, and they would be basically sort of hidden in water, right? So now proteins, if you remove the water molecules, proteins actually are close packed solids, right? We can define them as close packed solids. Close packed means they are dense and they are together, and they are based on these kinds of close packing globules. All right. So these proteins are coming together in the form of closed packed structures or collections of these solid globules. This is kind of hard to see what's happening here. So very often what you do is, instead of looking at all the side groups of amino acids, you specifically focus only on the backbone of the proteins. All right. So you switch off whenever you're looking at whenever you're looking at protein structures in 3 dimension, you switch off the representation of this ball and stick model, and you switch off the all the side groups, and you only focus on the backbone. So this here is only showing you the peptide bonds of between different amino acids, and this helps you. This backbone or main chain representation can help you understand the underlying relationship between different amino acids. So what do I mean by that? So for example, if you look at something like this here, it shows you that the peptide backbone is basically folding like this, and probably these are this is the structure that is basically interacting. This looks like an alpha helix. Similarly, this part of the protein is folding like this, and it's forming this beta sheet structure. So the backbone and main topology representations can help you not only talk about the larger structure and how amino acids are coming together, but if you zoom in, you can even go and reveal secondary structures. Now, because you can go and reveal these secondary structures, you can actually impose the secondary structure representation onto these backbone figures. All right. How do you do that? First, you just get rid of all these molecular representations and you only show carbons, right? You connect all the carbons together and you make what's called a tube representation. This is the simplest view of proteins, right? Next, you remember we showed zoomed into this part of the protein and this part of the protein. Now, do you see the second structures emerge? So this part of the protein actually looks like a alpha helix, right? And this part of the protein
Yes,
actually
sir.
looks like a beta sheet. So now you can impose these predicted structures onto these tube structures. So here, all the purple highlighted regions are alpha helices, and these yellow highlighted regions are beta sheets. Now, in order to make this easier to understand what these tools do, that some you will be having a very exhaustive lab on Wednesday, where you'll be looking at all these structures. What these tools do is they superimpose these predicted how these alpha helices are going to look like, or how these beta sheets are going to look like. All right, so you have a tube structure which represents the backbone of the protein, on top of which the predicted secondary structures are sort of put. All right, is this clear to everybody? How we are making up the protein structure in
Yes,
three
sir.
dimensions?
Yes, sir.
Yes, sir. So now you can now go not only this, not only you can see the secondary structure, you can also now see how the secondary structures are coming together and forming these kinds of tertiary structures. The next thing that you can do is these kinds of structures, they are dynamic, right? Therefore, whenever researchers are finding these structures, you will find that the same protein might have multiple structures. So when you align these structures one over the other, you can get this kind of a dynamic picture of how this protein is actually flexible, right? So here you can see that this part of the protein seems to be quite flexible, right? It's moving in this direction. This part of the protein seems to be very, very flexible. It's moving all around. But this part of the protein seems like even after overlapping many structures, they follow almost the same tube pattern, right? So viewing multiple superimposed structures solved under different conditions, let us say you increase the salt condition, you increase the lipids in your when you are crystallizing the proteins, you will get different kinds of structures, right? So you can then color the structures based on what's, say, active and what's inactive. So something which does not change after superimposition, that would form a part of your inactive protein structure, and some parts which are changing, they form your active part of the structure, and you can correspondingly color the protein based on this. All right. So all of these kinds of things you will be pursuing in the lab. I hope many of you do attend. All right. Then specifically, we can actually analyze that what motions in your these active structures, dynamic structures, what motions are very weird, what motions are like much more than you would expect just by chance or just by random motion of molecules. And then you get to specific parts of the protein which might actually be changing the protein structure a lot, and you get what's called things like these activation loops. So this part, you can zoom in and you can see that this particular part of the protein actually changes in a large manner. And if you imagine that top part and bottom part, it's almost like how shall I say, it's almost like a book. So this is a loop. So this loop can flatten out and this red part can move here and this blue part can move here and they will be further apart, right? So, and when this loop compresses, so the blue part comes here and the red part comes here and then they come together, right? So analyzing these multiple structures actually reveal some functional motions or displacements that are or could be important for regulating the function. Now, would you test that? You may make mutations in this part of the protein, which stops this kind of loop by, I don't know, adding like a proline, right? Proline essentially breaks. I hope everybody remembers what proline does. It sort of constrains, right? Because there is a the weird way in which there's a ring that forms when you have a peptide bond. I hope everybody remembers that prolines are called helix breakers. Yes, no,
Yes.
right? So you can make mutations like that and you can stop the motion and then you can see whether the protein is still performing its function. All right. So the key concept again was this whole idea that you have your unfolded protein, which forms this semi-compact molten globules, which then have try to have more conformations and which change and get even more compact to form what's these native conformations, which you can have multiple native conformations. And actually, when we talk about these multiple native conformation, this is what we are talking about. So this, you see this activation loop here, right? This activation loop gives you one kind of a shape. So the protein has one kind of a form. And then when the activation loop is sort of flattened out, then the protein gets sort of flattened out and probably becomes inactive and you get another conformation of the protein. So the large part, so these two parts of the proteins with respect to each other, they don't change. But when the activation loop or these kinds of loops in the proteins are changing, are changing the structure, then there would be large structural changes in the protein, which one can actually predict by looking at the structure. So now, what are the key forces that are affecting structure? There are four. You have hydrogen bonds, you have van der Waals forces, you have electrostatic forces, and you have hydrophobicity. So I think everybody understands how hydrogen bonding works. So you essentially have donors of hydrogen bonds and you have acceptors of the hydrogen bond. And typically you would form bonds between hydrogen and nitrogens, hydrogen and oxygens, right? And based on the angle, you would form a specific kind of a interaction. So whenever we're talking about these structures, these are the forces that we are trying to see how these forces are changing and how by changing the shape of the protein, uh, some free energy is released by stabilizing the structure through these key forces. So hydrogen bonding is one. Van der Waals forces is another one where you basically have a repulsion based on how close the molecules are, because when you have these structures, you see this kinds of solid sort of surfaces. They essentially represent the inability of the protein or protein molecules to go beyond the van der Waals forces. So this is how van der Waals forces would be affecting. Then you would have electrostatic forces, say, for example, between carboxyl and the amino group, or on the same peptide or on different peptides, which leads you to formation of the secondary structures maybe. And they form things like ionic bonds or salt bridges, which is determined by these Coulomb's forces. And you also have hydrophobicity, where the protein itself or the particular amino acid, there is like phase separation, where proteins which are having more hydrophobic residues or polar residues, they would try to basically segregate, right? So the molecules will aggregate together rather than completely dissolve in water based on the hydrophobicity. All right. So whenever we are talking about
Then apart from that, there are sulfide bonds also there.
sulfide bonds are there, but sulfide bonds are covalent bonds, right? So once the sulfide bond is formed, it will be formed. It will not be a dynamic property.
Yes,
So
sir.
here
Yes.
we are talking about dynamic properties. So you fold the protein differently, right? You have these two forms. So all these things will change. But if you have a disulfide bond, right? Let's say there was a disulfide bond between these two parts of the protein, then the protein is not going to be flexible, right? It's going to be constrained. So nothing will
Yes,
change.
sir.
So I request all of you. So we have for a few minutes. Please go to this website rcbsp.org. This is how it looks like. This is the protein data bank, which is where most of the structures are deposited, and we can go deeper into it. Let me see. I had opened this somewhere. Yeah. So once you go into, can you see my screen? Probably you cannot see my screen, right?
We can see. Now we cannot.
Can you see my screen? So now what you can do is you can go into this PDB bank, and let us say we want to look for PDB structures of proteins which are coming from the SARS-CoV-2 virus, right? Specifically, let's look for search for the spike protein of SARS-CoV-2 virus. All right. So when you click on it, it's going to give you all these different structures. So the same protein has been solved multiple times, right? Sometimes it is the alpha variant, sometimes it's the beta variant, right? Sometimes there's the mink variant. So mink basically is you had in Scandinavia a lot of these minks or mammals which are used for making expensive fur coats. They had an outbreak of virus, right? So a lot you have different conformations. This is a closed conformation. You have this is an erect conformation. So there are different ways structures which have been solved based on different kinds of conditions that the protein was isolated in, right? So now let's, for example, click on one of these structures. Let's click on this first structure, 7BM. It's a closed conformation of this variant of the of SARS-CoV-2 protein, right? And this is how this website looks like. It's going to give you, let's say, some information of the Ramchandran plot. It's going to give you some information about what folds this protein is having. So, for example, this is your protein, right? It gives you
Yes,
information about the
sir.
hydropathy of the protein. It gives you information about whether this protein is folded or not folded. It gives you information whether this protein is a part of some protein motifs coming from the PFAM, right? But this is something that probably you can explore later. We'll be exploring in your lab as well. But what's more interesting is if you click on this structure, you will get to see the protein actually in 3 dimensions along with its structure. So for example, this is how it looks like. And you can now click on it, this structure, and you can move it around. So see, this is how the spike protein is actually looking like in 3 dimensions. You can specifically go to regions. So let's say I go to the regions here, and you can see that we are moving around in this specific part. Can you see? So we are moving in this particular loop of the protein. Similarly, if you are, I'm at this alpha helix, right? Let us say I want to see exactly what structures are there in this alpha helix of the protein. Give me a second. It's kind of slow. So let us say I go here, right? So the area corresponding to this particular protein will light up. So let us say I click on this. I can click on this, and now what it does is it now also shows you the hydrogen bonds, right? It's also showing you the side chains, not just the backbone only, right? So this is what you'll be doing in the class, and this is what one important part of structural bioinformatics is to be able to visualize these proteins and try to derive information from this as I showed you. Okay. Can I
Yes,
see a
sir.
show of hands? How many of you were actually able to open and look at the protein structures?
Oh, sir, I did not.
So I'll wait for you, Ratnesh, and Deepak.
Oh, sir, actually, I forgot, like, what did you search there?
I searched for spike protein of the SARS-CoV-2 virus. Okay, so in the next class, now given that we have understood or have some basic idea of how proteins you can look at and how uh, their structures can be understood, in the next class, I'm going to talk about how different potential functions can be used to describe the energy of this protein structure. And so there are two main ideas. You can either use physics or you can use prior knowledge. So when we talk about physics, it would actually be some information about bonds, how bonds work, how bonds can be stretched, how they can be rotated. And when we talk about knowledge, it would be more based on some prior learned heuristics or observations. And these things can be used to now not only look at protein structures in static way like this, like I've showed you, but you can now, by applying these principles, you can try to understand the dynamics. It's called the molecular dynamics. So this is another specific field of structural bioinformatics, is to understand the dynamics of proteins, or molecular dynamics of proteins. So for example, Professor Sundar in our department is an expert in molecular dynamics of proteins. All right, I'll stop the class here. I'll take questions if you have any. Please keep your hands down and raise your hands only if you have a question.